So today's presentation will be talking about cell reproduction. And I'm not talking about cell reproduction, not reproduction in a sexual kind of way, but mostly reproduction at the cellular level. So what I mean by that is I want to explain how cells go from one cell to two cells to 4 cells and 8 cells, etc. So this happens every time an embryo develops. We start with a very small amount of cells, and these cells will grow and eventually will form different parts of the body, organs, and etc. But it also happens when we get an injury. So if you cut yourself, for example, or if you have parts of your body that are damaged in some way, some of those parts may be reproduced, may be repaired, and cells may after that occupy a space they didn't occupy before. So for example, we know that we can remove a sizable amount of your liver, and that liver, if it's cut to a certain size, has the ability to regenerate itself almost completely. So this can only happen because cells are allowed to grow again. So they divide and grow and divide and grow. And this happens every time you get a cut or some kind of a scratch or something. When that happens, the cells are scraped off, and of course that exposes the tissue underneath. Well, in order to repair that tissue, the cells will have to divide on top of it and then recover and then protect the tissue again. So we need those cells, those skin cells, for example, to grow back to where they were and how they were before in order to protect the wound. So this can only be due to mitosis. Unfortunately, when we're talking about mitosis, we need to understand that cell reproduction is a highly controlled series of events, and of course at some point this can lead to the development of cancer. This is not something we are going to discuss right now, but please keep that in mind, is that the cells, when they're dividing, they need to make sure that they're following a specific plan. So what we have here is an embryo, and I said that I wouldn't talk about reproduction in a sexual kind of way, and of course this is not my goal right now, but what I want to show here is that the embryos, as we see here in part A of that slide, what we have here is the envelope of the embryo right here, and then what we have inside right here and right here underneath are 2 cells, right? So we have those 2 cells, and those 2 cells will divide at the same time and will cut in half, both of them, to lead to part B here. So you see the embryo now started at 2 cells right here, and now it ends up at 4 cells. So after one cycle of cell division, we end up with a 4-cell embryo. And then as this goes on and all this division for a while is synchronized, we will move on from 4 cells and then to an embryo that now has 8 cells. And then so on and then so on. And of course this leads to more complex division and development after that, and then we can see it here. So for example, again, if we're talking about an embryo, we have the moment of fertilization, and then the cells will accumulate and move on from 2 cells to 4 to 8 to 16, 32, etc. And then at some point we can see that the embryo gets more and more and more complex, as we can see here, right? It's much more complex, and all of this here we have throughout all of these processes as they go. All of these processes actually is going in the other direction, but it doesn't matter. All these processes here will involve a higher number of cells. So in this case here, that part of the embryo has way more cells than what we had here, and then we have way more cells here. So in order to create an embryo, we need some kind of synchronization between the cell division. The cells need to divide at a certain time, they need to stop at a certain time, and then we need to restart again. And that's how an embryo eventually is formed. So what we have here is the representation of a graphic representation of what the cell cycle is. Okay, so here's what we have. We have different parts of the cell cycle. So most cells in your body right now are at interphase. Okay, so we say G1, they're at interphase. So basically what they're doing is that they're resting. Nothing much is happening. They're maintained alive by consuming oxygen and consuming nutrients, including amino acids, sugars, etc. And they're not necessarily dividing. This is why you maintain a certain weight. This is why you maintain your height throughout most of your life. So basically, most of your cells are in this phase. But eventually, many of those cells, they need to be replaced, right? So your body will lose some cells and will replenish some cells. Just to think about your immune cells right now, right? So you have many of your immune cells that are being divided and created. At the same time, you have a lot of those immune cells that are dying and being replaced. Okay, so what that means is that some cells will die, but also some cells will be born by a new process. Now, the new cells that are being born, the new cells that are being formed, will need to go through the process where we have one cell, and that one cell eventually is going to divide into two cells. We say that those two cells are daughter cells. So we have one cell here, and then we have two daughter cells right here. And those two daughter cells eventually will divide into two more cells, and then we'll have at the end, we'll have four more cells, provided that the process is allowed to continue. So in order to go from this step here to this step here, we need to follow a set of very interesting and important instructions. And then what we need to do is we need to go through the cell cycle. So because we know that the cell here, the mother cell, if we can say, has a certain amount of DNA, and if it's a human cell, we know that a human cell has two sets of 23 chromosomes, two sets of 23 chromosomes for a total of 46 chromosomes, right? 46 chromosomes. So we know that this, for human cells, that's the number, the total amount of chromosomes we have. So when we go from one cell to two cells, right? So from one cell, and then we end up at the end of the cell division, we move on to two cells. Well, those two cells here, they need to be viable. So basically what we need is a cell that starts with 46 chromosomes, but then at the end of the cycle, at the end of the cell division, we need to have two cells that also have 46 chromosomes. So if you count well, 46 plus 46, that's a total of 96 chromosomes. So in order to 92 chromosomes, not 96, 92 chromosomes. 92 chromosomes. So in order to end up with 92 chromosomes and then divide this into two sets of 46 chromosomes, you can imagine that the DNA that's found inside that cell here will need to be replicated. We will need to have twice the amount of DNA before we even start dividing. So that's what we see here when we're thinking about DNA synthesis. This is what that is here. So when we look at different types, when we go through this cell cycle, we go through a phase of interphase, and then we move on to DNA synthesis, which is known as the S phase, and the S phase, of course, for synthesis. Then once we end up at the end of the DNA synthesis stage, we have twice the amount of DNA. So we started at 46 chromosome right here, and it started here. But as we go through this cycle here, at the end, we will end up with 92 chromosomes, right? Because we're ready at this point to go from one cell into two cells, two exactly identical cells. And in order to have viable cells, we need to have the right amount of DNA, which is, of course, 46 chromosomes, right? So before we even start thinking about going through this stage right here, which is cell division, we need to think about synthesizing our DNA. So we have, at some point, 92 of those chromosomes here, 92 chromosomes, and then we end up at the end with 46 chromosomes each. Okay, so then after that, while the cell has now divided its own DNA, it will go through a series of steps that will lead to something that we call mitosis. Okay, so mitosis is the action of actually dividing that one cell into two daughter cells. But before we get there, don't forget, we need to have DNA synthesis. We need to have enough DNA to provide for those two new cells. Now, before this even happens, before we even get to mitosis, we get into another part of the cell cycle that's called G2. Now, we will look at what happens at G1S and G2 in this presentation, but I just wanted to, make sure that you understood what the sequence of event that's what's happening here. So soon we'll be talking about cell division and how DNA and chromosomes are being split up between the daughter cells. Before we even start doing something like that, I really want to explain the concept of DNA and how DNA is organized into our cells. So we know that in our cells, the eukaryotic cells, we have a nucleus, and we know that inside that nucleus, this is where we have the double helix, right? So we have our DNA in that nucleus, and that DNA, although we often think about DNA, we think about the double helix, and we think the double helix is like this, and that's totally true. That's the double helix, and it's connected. The two elements of the helix are connected between the two of them via what we call hydrogen bonding. So that molecule is always, it's always true that this molecule looks like that. However, there are many steps in the way DNA is organized inside a cell. So just think about that, right? So in one cell, in only one cell, it's tiny. We can only see that cell through the microscope, and we have an even smaller cell nucleus, and all of that DNA is compacted inside that cell nucleus. So just think about the fact that when we're looking at all the pieces of our DNA inside a cell, which is 46 chromosomes, if we could put all of these DNA pieces one after the other and measure them on a table, all of those pieces of DNA one after the other would measure approximately 1 meter, right? So that's a lot of DNA in a very, very tiny organelle, which is the nucleus. So how does the cell do that? Well, the cell is organized. The cell is always organized, especially eukaryotic cells. They're highly efficient, and they do everything with organization. So this is what we're going to look at in the next few slides. So it will go from this molecule here, which is the double helix, but we'll go through a series of steps of organization that will lead to eventually the appearance of the this mitotic chromosome. Now let's start with this. What we have here is our double helix, and as you can see here, right in the middle, right here, everywhere, we can see that the double helix is maintained together by a series of interaction between the two strands, one strand like this and then the other strand like this. And what we see is that these strands are maintained together by the interaction right here, right in the middle, of molecules that are integral part of each strand. So what maintains those two strands together are hydrogen bondings. Now, when we're thinking about the nucleus, and remember that the goal here is to try to fit as much DNA as possible in a very, very small part of the cell, which is the nucleus, well, we need to organize this a little bit better. Now, I'm going to give you a metaphor here that I think is going to do the trick in explaining how this DNA is organized. You know, when you have a hose in a garage, right? So you have a hose somewhere in your house, or somewhere around your house, and this hose can kind of randomly found on the ground, and of course it creates knots and everything. So whenever you're trying to use that hose, there's always a few knots here and there. It's very annoying, and you need to undo it and try to make it like as straight as possible in order to use it. Well, there's a way to make sure that this hose is organized at all times, and we can organize this hose around a barrel. We have these barrels like this, so where we have the two sides, there's usually a handle here, and then right in the middle here you'll have your hose that will be wound around this barrel right here. So we have this hose like that. Well, in order to organize this DNA, we have a bunch of molecules right here in the middle. You see those molecules here in blue? Those are called histones. And histones bind to DNA. They bind to patches of DNA here and there. And what they do is that they wound DNA around themselves. They organize this DNA around themselves, and they create those loops. And of course, there will be many of those loops all along every single chromosomes, right? So we have loops here. So you see in blue, we have the histones. So basically, histones are just a bunch of proteins that have the ability to interact with DNA, and it wraps DNA around themselves. Now, what's interesting is that each of those histones have the ability to interact with each other. Okay, so what we'll have is our DNA in red right here. So we see the DNA right here in red, and then in blue right here we have the histones, right? And those histones are interacting with one another, and they create this mega spiral. So just think about DNA, and at the basic structure of DNA, the double helix like this. Now the double helix it's still found here. We can see it here in very, very small portions, but now this double helix is organized in itself in another helix. Okay, so if we could take this molecule right here and look at it from the top, basically what we would see is the histones like this. The histones will be distributed like that. Okay, so we would have 6 histones like this. So this is the part that's blue, and then DNA would be wrapped around those histones like this. Okay, and they would form this long stretch. It would go in both directions and would be organized like that. So as we can see here in this second part of that slide, we have our DNA that is organized in a long stretch, and that DNA is wrapped around all of those histones. So it's really, really very well organized. Now, because the cell is, again, super organized, these histones here will interact with another type of protein that are called scaffolding proteins. So basically what we see here is that these are the stretch, right? What we see here in blue, all of this here are the histones right here, as seen in this part right here. But what we see is that those histones are now organized around other types of proteins. So basically this part here, this helix of double helical DNA, if hopefully that makes sense to you, is now further coiled around scaffolding proteins, and we can see this here. So now what we see is a super helical structure. So DNA is really now compacted around those molecules, right? So it occupies a much smaller space. Just imagine how messy that would be if we didn't have those proteins here to take care of the organization of DNA. And of course, that leads us to a extreme organization. I don't have enough word to express how organized all of that is. And that DNA is organized like this in spiral around furthermore scaffolding proteins. And then at some point, and this is seen only during cell division, okay, so that's very important. It's only during cell division we will see those chromosomes like this. They have like this kind of X structure, and what we see here is an actual chromosome. Is a picture of an actual chromosome, and we see that we have 2 parts of this chromosome. We have one part here, and then we have another part right here, and those are called sister chromatids. Okay, so sister chromatids. Sister chromatids right here. So there's one chromatid right here, and there's another one chromatid right here. And then what you can see here, there's banding, a banding pattern that we see. That's very typical of mitotic chromosomes. We can see the banding pattern, and the reason why some spots are darker than other ones, so you see this part right here is much darker than that part here, it's because there's the winding is much tighter in that region. Now, the one thing that I want you to remember is that DNA inside a cell, so this is my cell here, and then I have my nucleus right here. My DNA, well, the DNA of the cell, will be organized in some way, and what we've seen earlier, all those patterns here, these patterns like this, well, depending on the state of the cell, depending if genes are needed and if DNA is active or not, depending on that, we will have different states of organization, right? We will have states like this one, or maybe like this, or maybe like this. But of course, when the chromosomes are formed, this is the only time when we find these chromosomes here, okay? So when you look inside a cell and you look inside the nucleus, as we can see here, that DNA will be at some point much denser than other spots. So you have patterns in there. Some part of the DNA is much tighter, and so you see the coloring of the inside of a nucleus is not homogeneous. It has different parts of darker and lighter areas, and this is why we say that this is part of that nucleus and that DNA is called chromatin, and then what we see here is part of that chromatin, right? Chroma meaning color, right? That's where this word is from, chroma, and it means that inside the nucleus we see pattern of kind of like coloring, not that they're colored in any way. If we look at it through the microscope without any dyes or anything like that, most of the time that will be gray, black and gray like we see here, but we can see patterns of DNA organization in some spot. And now remember that some part of this DNA will be less organized, less wound up around a packaging of histones and scaffolding proteins, and some other parts will be highly organized, right? So it's not homogeneous. Now, when we look at cells that are ready to divide, when cells have gone from G1 and then they've gone to the S phase, and we know that the S phase, if we have n amount of chromosome at G1, then at S phase we will have n times 2 the amount of chromosomes, right? So we had, say, at the end of S, so at first we said, okay, we have, say, 23, so we have a total of 46 chromosomes for human cells at G1. Then at the end of that S phase here, what we need to do is divide all of that DNA and multiply it basically, and then we will have 46 times 2, which is equal to 92 chromosomes, right? So at the end of S phase, we will have 92 chromosomes. So cells, when they're supposed to divide, they will go through different things. Now again, cells are always following a plan. They just don't divide without a plan. Everything needs to be organized. Everything needs to follow that plan so that everything is done properly. Just imagine if cells could divide without having the ability to divide, because chromosomes are not distributed correctly, or because your DNA is not synthesized, and there's not enough DNA for 2 cells, etc. That would lead to, of course, catastrophic consequences for the cells, right? The daughter cells might not be viable, or even worse, if they are viable, they can lead to disease. They can lead to problems with the rest of those cells after when they divide. So everything needs to be as clean as possible. So the cell will go through the process of dividing, and that process is called mitosis. mitosis. They'll go through this process, and we'll, at the end of mitosis, this is when we have 2 new daughter cells, and only at the end of mitosis. Now, the next few slides will deal with the concept of mitosis. We will look at what's happening during mitosis between the time when we have, at the start, when we have 2 N chromosomes, or in our case here 92 chromosomes, and at the end of mitosis, when we have 2 daughter cells that each have 46 chromosomes. Okay, so the next part is really dedicated to that. Now, when we look at a cell, we see that the cell has a certain structure. So we have our cell here, and then we have the nucleus, and most of the time we can see the cell looking like that. Okay, so if you take any of your cells right now anywhere, if you take like we did this in the lab, when you look at your cheek cells, for example, well, all of those cells, or for the most part, would be like this, kind of round or close to that, and then you would see the nucleus on the inside. So we use methylene blue, and methylene blue allowed us to see the nucleus right here. So those are called interphase cells, when the cells are in between divisions. So that's called interphase. So during interphase, the cells really roughly look like that. Some cells are round, some other ones have other kind of structures and shapes depending on the cytoskeleton. But basically what I'm doing here is just drawing a generic cell just to explain what we see here. Now, what's interesting is that the nuclear membrane, so the membrane that surrounds the nucleus, where the DNA is on the inside here, is a membrane that has the capacity to disappear. It will be disassembled by the cell as the cell proceeds through the mitosis cycle. So what we see here is the first part of that mitotic cycle, and that's called prophase. We'll see that there are different types of phases, but basically they go with the acronym PMAT. I like to say PMAT. So prophase, metaphase, anaphase, and telophase, and we will look at every single one of those phases and what it means. So coming back to prophase, what we see here is that, remember that chromosomes and DNA inside a cell during interphase, we can't see the X's, right? We cannot see this if this is a nucleus here. We know that the DNA is organized into chromatin, and some part of that chromatin is denser than other parts. But yet we cannot see still the chromosomes, the individual chromosomes inside. And that's because they are in a state, we say, they're not condensed yet. Well, when we look at the first phase of mitosis, which is a prophase here, we see that the chromosomes will start to become visible because they will condense. So inside that nucleus here that is now starting to disappear, we say that the nuclear envelope breaks down, and we can see it right here. The envelope is actually breaking down, and at some point will disappear. Look in prometaphase, we don't really see the nuclear membrane anymore. So what we see here is that we have the nuclear envelope that breaks down, and that's normal. We don't want this shell around the nucleus. We want to get rid of it, because what we want to do is separate that DNA, right? We want to create 2 daughter cells, and the nuclear envelope will get in the way of that. So this is the first part. What we have here is prophase. What's interesting as well is that there's 2 organelles in the cell that are known to be, they're called centrosomes. And if you remember when we presented and talked about the different types of proteins we have in a cell, such as the cytoskeleton, we saw that a cytoskeleton is made of different types of proteins, including microtubules, right? So what we have here is the centrosomes, and out of the centrosomes come the microtubules. So you can see those blue fibers here emerging from this inside here of that centrosome. We see that those fibers are emerging from that. Now, when we look at pro-metaphase, just before metaphase, so we looked at prophase, now we will be looking at metaphase, but before we look at that, we will look at pro-metaphase. So basically it's a phase between prophase and metaphase. So the chromosomes will further continue to condense, so we'll see them more and more and more. So they're really organizing themselves around this scaffolding protein. And what we'll see that is this. We see the kinetochores will appear at the centromere. So what is the centromere? Well, the centromere is this. When you have your chromosomes that kind of look like this, right? They have this X shape like that. What we see here, the kinetochore, is something that is found right in the middle here. Okay, we see this here, those two. So what is contacted basically by the microtubules right there is known as the kinetochore. So the kinetochore will appear at the centromere of the chromosome. The centromere is the region which is the most central to the X shape. Then the mitotic spindle microtubules will attach the kinetochore. So you see here that we have some of those microtubules that are actually making contact with the kinetochore. We see this right here. Okay, so we see this right here. We see another one here, another one here. Okay, and then what's going to happen is those that are contacted, those microtubules will at some point start another chromosome, sorry, the centrosomes will move at the pole. So you see they started here somewhere in the cell and they'll move at the pole. Why are they doing this? Well, because we want to divide the cell in two, right? So in order to be doing this, we need two machines, basically, two biological machines that will be able to attach the chromosomes at the centromere via the kinetochore, which is part of the chromosome right here, and they will need to pull apart pull those chromosomes in each direction. Now let's move to metaphase. So metaphase is when we have a movement of the chromosomes on something that we call the metaphase plate. Metaphase plate right here. So you see all the chromosomes will be aligned. Now remember that for human cells we have 23 times 2 chromosomes, so 46. But then at the end of the S cycles we have 92 chromosomes per cell, right? So that we would see here. Of course we cannot show all of those chromosomes here. We're not interested in doing that. So what we show here is only 4 chromosomes, but remember there's way more than that. So what we have is all of those chromosomes, and those chromosomes will now be aligned on a plate. And this is just prior to cell division. Now when we look at electron microscopy right here with our DNA being colored in blue and then the microtubules being colored in green, we can see that the chromosomes are aligned perfectly on the middle of that cell. So remember that when we're looking at the mitotic cycle or mitosis, we have P, M, A, and T, and then we have this little part right here in the middle. So we have prophase, metaphase, anaphase, and telophase, and then we also have pro-metaphase right here, which is in the middle of those two. Now we're going to move on to anaphase and then telophase. Telophase is the end of it. It's the end of those phases. It's when the cell is just about to divide, and that's just a phase just before that. Now let's look at what's happening here. Now we are into anaphase. What we saw earlier was the alignment on the metaphase plate right here. The chromosomes were all aligned here. And then what we see now is that we can tell that those two sister chromatids that were held together right here, right at the centromere, now are being detached. So what we see here is that those chromosomes here are now being pulled apart in each direction. Okay, we see that. So those microtubules originating from the centromere right here, originating from the centrosome, are now being pulled apart this way in this direction here by the microtubules. So we have the separation of those microtubules. And if we looked under the microscope, we would see this here. So there's a mistake in your book, and the mistake is this, is that what we see here is basically the chromosomes being pulled away, and we see this. And then at some point the cells will lead to telophase, which is the complete separation of those chromosomes. So that's at the end. So what we have here is really the separation of the chromosomes. Okay, so telophase, the nuclear envelope at the beginning of telophase will start to reform again, right? Before even we start to divide the cell, the nuclear envelope will reform, and that DNA will start to decondense. Okay, will begin to decondense. So basically, the X shape of the chromosome now disappears, and it starts to be decondensed again and form chromatin. At the end of telophase, now it's time to produce two daughter cells, and those cells will divide in the middle here, and that process is called cytokinesis. This is when the cells will separate. Now there's a little bit of difference between animal cells and plant cells. When we look at animal cells, there will be a cleavage furrow that will, be formed here. So it will pinch the middle of those cells into a very, very small area. So it will look something like this. Right? So we'll have our two new nuclear membranes, envelopes created with the DNA on the inside. And this here eventually is going to pinch and lead to the formation of those two daughter cells with the nucleus like that. In plant cells, it's a little bit different because plant cells usually have a shell around them, some kind of a cell wall, and the nucleus is still inside, right? The type of telophase and all of these phases still happen in the plant cell, but at the end there will be a little formation of a wall right here in the middle to separate the cells, so that all of those cells after that in the plant are still surrounded by that cellulose shell or cell wall around them. So again, in electron microscopy, well, actually, it's not even electron microscopy, it's just microscopy, but what I meant to say is that it's fluorescence microscopy, that's what I meant to say. And then what we see here in green are the microtubules, and what we see here in blue is the DNA, okay? So that DNA here, and then we can see that in prophase, that DNA is starting to condense, okay? So we can tell something's happening here. We can see that parts of that DNA is now much, much denser than the chromatin it was before. And we see all of those microtubules here that will start to organize in a certain way. Now, what we have here is the centromere, and out of the centromere, we have the spindle that we call, and those are the microtubules. And the microtubules will attach to the kinetochore. So you see those spots here in pink, those spots here are the kinetochores. Okay, so we don't see the chromosomes yet. They're still condensing because right now we're not in metaphase. We are in metaphase. So that's that phase in between prophase and metaphase. Okay, so pro-meta phase, and then we can still see this DNA here being condensed more and more and more. Now remember that DNA is colored in blue, and then what we have here is our microtubules here in green, and then we have a very neat arrangement right here and a metaphase plate, right? We can see all the chromosomes are aligned and attached to those microtubules, and they will be ready to be separated into two poles of that cell that is not divided yet. So this here is the metaphase. And then as we move on, the cells, the microtubules will divide and separate the DNA into two parts of that same cell. So we are still in one cell. What happens here though is that the DNA now has moved on to a separate part. So we have the two sister chromatids that are now separated. So we are in anaphase here, anaphase right here, and then we have the two parts of our DNA that is now separated into two regions, and eventually, of course, this will lead to this part here, and then the cell that at the end of this phase, which is telophase, the cell will actually form something like this, a cleavage furrow right here in the middle, and this will be cut, and then we will have our two cells. At the end of telophase, so this is not cytokinesis yet, this is not the division of the cells yet, but I'm just moving ahead to show you what's going to happen. And at some point, the nuclear membrane, those chromosomes here are going to decondense and then form chromatin again, and the nuclear membrane will reform, so that at the end, of course, we have our two daughter cells that have, each of them have the nuclear membrane and of course the DNA inside. So what I want to do here is come back to that cell cycle, and I want to talk about the control of the cell cycle. I want to talk about something very important, because as we know, the cells, it's important that they divide. It's important that they go from phase G1 to phase S, and then eventually phase G2. And of course, at some point, they'll go to the mitotic phase right here and form two daughter cells, right? So we have one cell leading to, of course, two daughter cells at the end of the whole process. Now, we want those two cells here, this one and this one, those two cells, we want to be as healthy as the mother cell right here. So we want these two cells to have the same amount of DNA that the first one had. And that's very, very important. So in order to do this, the cell will check the status of this as it goes along. Okay, so as the cell proceeds to all of those different phases, the cell will check and see, well, okay, so where are we at? Are we ready to go? So the first one here is the G1 checkpoint. Okay, so we also say, a restriction here. So what's going to happen is that the cell is going to decide there's either a go, right? So we're ready to go, or it's a no-go, right? Either we decide we commit to this division and we will synthesize DNA and then eventually we'll divide the cell, or we stop and we don't do anything here. So the cell here at this G1 checkpoint will make sure that all the conditions are met to allow cell division. And the question, if the cell could talk, would say, do we commit to this or not, right? So one of the questions could be, well, are we big enough right now? Do we have everything, all the proteins we need? Do we have all the nutrients? Are we healthy enough to go through this cycle? That is absolutely crucial. And another very important question would be to ask, okay, well, is that DNA that I'm supposed to synthesize, this new DNA that I'm supposed to create, well, the first part of that DNA, before we even think of synthesizing that DNA, is this even damaged? What if there's some damage? Can we repair it? Should we repair that DNA? If the answer is yes to all of those questions, of course the checkpoint will be gone. We'll go through that checkpoint and then we'll be ready to move on to DNA synthesis. Now, once this process starts, it doesn't stop. So the cell will do everything it can to synthesize its DNA, and it will do so, without problems usually. And then the cell will grow. Okay, and at some point, even before moving to the mitotic phase, so which is dividing the cell into two daughter points, we will go and the cell will stop again at another checkpoint that is called the G2 checkpoint. So what we want to ask here is that before we even get into the M phase, we need to ask, okay, well, that DNA, all of that DNA that was synthesized here, all of that DNA, is it intact? Is it repairable? Is there any damage to it? And if there is any damage, is it worth it to repair it? Sometimes the cell will decide, no, it's not worth it. I cannot repair that. It's too much energy. And then that cell goes into some kind of a process called apoptosis, where the cell basically decides to close the shop and then it dies, right? But it's a program, it's called programmed cell death. So the cell will basically kill themselves in order to not propagate any mistakes that could be part of that DNA. So you see, all of those questions need to be answered before we do so. So at the end, when this checkpoint is gone, so we say, okay, yeah, our DNA is we have enough DNA. We have twice the amount of DNA. It's not damaged, and if it was damaged, then we can create. We can repair it, so it's worth to repair. Then and only then we will move on to the mitotic phase of that. But hey, guess what? There's another checkpoint right here. So what we want to do here is we want to verify that as we go through all those phases here, PMAT, including prometaphase, all of those spots here, we need to verify that everything is okay, especially at the metaphase spot, right? Remember what happens in the metaphase. We have our cell, and then all the chromosomes are organized on the metaphase plate right here, and then on each side right here, we have centrosomes that are attached through microtubules to the kinetochore of that chromosome here at the centrosome of that, at the part of the chromosome here. So the centromere, not a centrosome, but the centromere. So we have the centromere here, and we have the microtubules are originating out of the centrosome. And I know it gets confusing, centrosome, centromere, and all that, right? But anyway, we need to verify that everything is okay here, and that there's no DNA or chromosomes left behind, because we want to separate all of those chromosomes, right? So in that case, we need to ask the questions. We need to answer those questions. So we would say, well, are the sister chromatids correctly attached? Are they really attached to all of those microtubules? That's very important. And are we ready to pull on them? Are they firmly attached? Can we pull on those cells so that we can divide those two at the anaphase and telophase, and at some point after that we'll go to cytokinesis, where we have our two cells. So all of those questions have to be answered. So all in all, what we've seen right now is the process of cell reproduction and all the phases of mitosis. Now, all of those phases are super important to understand. I'm not asking you to understand the molecular parts of that, but I want you to know which phase is, what phase are, and then what they do, and why it's organized in this way.